My name is Dane Comerford and I'm Head of Public Engagement, part of the team that puts together the annual Cambridge Science Festival. We've been on the Biomedical Campus, as you probably know, since 2014 as an effort to explore clinical, business and academic healthcare research. So I'm not going to talk much longer apart from introducing Professor Daniel St Johnson.
I'm here to introduce John Gurdon, and I guess normally on such an occasion one would give a potted history of John's career. But actually John is going to talk about his career, so I thought I would do it slightly differently and give my personal recollections of my interactions with John over my career. John started out in the other place, in Oxford, but he moved to Cambridge in 1971, first at the MRC Laboratory of Molecular Biology, and then as a professor in the Department of Zoology. And that's when I first ran across him, because I took this amazing final year course in developmental biology. And John's lectures impressed me particularly. One thing that struck me was that he would very quickly do these perfect drawings of complicated cell movements inside developing frog embryos. They were so clear and so perfect, I couldn't believe it was humanly possible to do it. Years later, I discovered John's trick, which is that he would go into the lecture theatre 20 minutes before the lecture and draw all the diagrams in blue chalk on a green blackboard, so it was invisible to the audience. And then, when he was in the middle of his lecture, he could trace out those lines extremely quickly and perfectly. And I think this attention to detail is one of the reasons John was such an inspiring lecturer. And John really led by example. Still, even though he was director of an institute, a university professor, a governor of the Wellcome Trust, still performing his own experiments several days a week. He would still come to all of the first-year PhD student talks and ask them penetrating questions. And he would even dress up for the Christmas party, and I particularly remember him appearing as Cruella de Vil one year, which was... The Nobel Committee decided to award John the Nobel Prize in Physiology or Medicine in 2012 with Shinya Yamanaka. When the prize was awarded, we all heard it on the internet. We went rushing around to find John, to congratulate him, and he was nowhere to be found. We eventually tracked him down. He was in a meeting room with his students and postdocs, and one of his PhD students was giving a lab meeting. And so we all gathered outside the door, and we wondered if John had actually heard the news. So eventually I was pushed to the front and knocked timidly on the door and went in. And I said, "John, have you heard? You've just won the Nobel Prize." And he said, "Yes, actually I did know. Some journalist called me up at 7 o'clock this morning." But of course his lab had no idea, and they were all cheering away and everyone was celebrating for about 3 minutes until John told us all to leave so he could get back to finishing his meeting. And I think that epitomizes John's character and his approach to science. The fact that he still works every day is just so impressive. But he is genuinely excited by what he can discover. So I'm very proud to be the director of the institute that bears John's name and that he founded, because he's been an inspiring mentor for me. And I think he has not only done outstanding science, but he's also fostered outstanding science. So without further ado, ladies and gentlemen, I'd invite Sir John Gurdon to come and tell us about the journey of a lifetime.
So first, thank you all for coming along on a Sunday afternoon. And of course, my first pleasure is to thank my head of department, Daniel, for his overgenerous introduction. And if there's anything I've ever done at all successfully, I actually believe it was to persuade him to join our institute. It's been an enormous benefit that he's running our institute for us. I suppose if you're starting out in life, hoping for a productive career, you might as well start at the bottom on the grounds that things can only get better. And that's, in fact, exactly what I did, not actually by intention, but that's what happened. Really traveled the world very largely on my school report. The schoolmaster said that Gurdon has ideas of being a scientist, and on present showing this is quite ridiculous. Can't learn simple biological facts, would essentially waste everybody's time. So that was the end of my science at school. And when that happens, of course, they take you off that subject and put you onto something they think you might do a bit better. Actually, I later discovered that they concluded that I had no ability to study any subject in depth. So I was then put on to Greek and Latin, not because I had any aptitude for that. It turns out the real reason was that all schools have too many classics masters. And so if you're pretty much bottom of the range, they put you into a kind of class with all the people who can't do anything better. So I spent the rest of my time at school studying Greek. And this might seem completely irrelevant, but it turns out it wasn't entirely so. It says that the eagle rose up, but the liver of Prometheus became immortal and grew every night by the amount that the eagle had eaten. And that's the story of Prometheus, who stole the story of fire from Zeus. And Zeus punished him by putting an eagle to eat his liver every day. This is a sculpture. There's the eagle, that's its head. It's got claws, and it's eating Prometheus's liver. And the story in Hesiod goes back to this 750 BC. And the key point is that the story says that the liver grew every night by the amount that they used a long-winged bird, meaning eagle, had eaten. Now that's awfully close to the real truth, because the liver does in fact regenerate, not every night, but somehow in 750 BC they somehow worked out that might happen. And this is the kind of earliest indication, perhaps, of the whole concept of regeneration. And that's the theme of this talk. I'll go back again a bit further back in history, because this is perhaps the first example of replacement of parts in the human body. This skull, or this lower jaw, has actually two false teeth. Those are made of stone. Dentists will tell you that these must have been hammered in real life. Pretty uncomfortable, because the bone has regenerated round those teeth. So that's probably the earliest example of people having replacement parts. Of course, there are current examples of regeneration. These are the deer, which you may know, the males regrow their horns every year. They fall off and they regrow this enormous structure. That isn't so much a replacement, it's just that the cells that make the horn continue to grow. So the same cells are involved, but they do this enormous amount of regeneration every year. Now, that leads me to the general theme of this talk. It shows a picture of an elderly person, and it's now possible to take skin from such a person and to do cloning or nuclear transfer to generate an egg and an embryo from the skin, an early embryo. That embryo can be grown into large numbers of cells, which are still embryonic, and then it's possible to add components to these cells and to make them form other structures, such as a heart or brain. And that's the theme that I want to explore in this talk. I'll say a word or two about normal development, then a bit about cloning, stem cells, and finally cell replacement. How do cells of different sorts, like brain and heart, how do they arise from an egg? You may be familiar, probably are, of frog spawn in the ponds around us. This is a mass of frog spawn. All of these are single eggs, but all of them can turn into tadpoles, which can swim around, and they do that without any instruction. The mother frog has long since gone, and no one tells those eggs how to turn into a living organism. So it's an amazing phenomenon that it can occur. The old ideas were that perhaps the sperm or an egg had a little man inside it, and this would gradually develop. But when they looked to see if there might be such little man inside a sperm or an egg, they could never find such a thing. And this is the picture, actually, of a frog egg, just the kind I showed you. Large single cell, and absolutely no indication of anything inside it that resembles what it's going to turn into. It's just a mass of yolk and cytoplasm. So the question is, how does that egg know how to turn into this complicated organism that it does eventually form? And I'm just going to now show you what happens when the egg does develop. These are frog eggs, and they're dividing 2, 4, 8, 16 cells. First thing it does is to divide into a large number of cells. This happens pretty quickly. About every half hour, the cells divide again. And they make a ball of cells like these, usually several thousand cells in a ball. When it's got that far, things start happening, and it will change. The cells on the outside move to the inside, make a multi-layered structure, and form the head and eventually the brain. And that is the head, and this is just the stage before the tadpole emerges from the egg and starts swimming round. That's how it actually happens in the case of the frog, and similarly in other organisms. So how does that happen? There are two fundamental ways we now know about how this can possibly take place. One is that the egg, although it looks like it's homogeneous, actually has different substances at one end, colored yellow, from those at the other end, colored blue. And when the egg divides into lots of cells, the cells at the top end have the yellow substance, and the ones at the bottom have the blue substance. So there are differences already arising. And soon after that, the blue cells send out signals to some of the yellow cells near them and tell them to become different. So this is the asymmetric distribution of components in the egg, and that is signaling between cells. Those are the two fundamental ways in which cell differences arise. The signaling is particularly interesting. It means that one end of the embryo will have a signal source of molecules which spread from there, shown in red dots, and these will reach cell C at a high concentration, cell B at a lower concentration, cell A lower still, and this cell doesn't get any. And according to the concentration of those substances, cell C will become something like intestine, cell B with a lower level of signaling molecule will become heart, A be brain, and finally skin. And that is the principle by which these different kinds of cells arise. As a result of that, our lives work this way. We start as an egg, embryo, and then a fetus, and eventually you have all these different cell types, which we're used to, like nerve and blood and so on. But the main point is that these differences between cells are extremely stable. Almost never does a nerve turn into bone or hair turn into intestine. Once the cells have settled on becoming something, they don't change. However, it is possible to make them change. The first point is that the cell differentiation, as we call it, is very stable, but it can be reversed by this process called nuclear transfer. And nowadays we call that cloning. Many of you may have heard the word clones. Cloning just means making identical copies. Identical twins are really members of a clone. And we ought to remember that when this work started in the 1950s, we did not even know that all the different cells in the body have the same genes. The heart and the brain and the skin, we know they now all have the same genes. Actually, that's necessary because if it weren't true, we could never do cell replacement. One takes skin, separates the cells, and then one cell is taken into a very fine needle which breaks the cell but not the nucleus. The nucleus is the pink area, and the nucleus is put inside an egg. The egg chromosomes have been removed, and in some cases this combination of the skin nucleus and the egg will form an adult animal, frog in this case, and nowadays the same principle has been established for a mouse and indeed other animals. These things are not as simple as you might think. First thing you have to do if you're trying to do an experiment like that is to make a machine that makes what you want. And this needle, when you break a very fine glass tube, it breaks irregularly, and it was necessary to make an instrument which will put a hypodermic tip like that onto the end of this broken needle. That all has to be done under the microscope, and you have to make the equipment to do it. But having done that, the experiment can then be made to work. When I started work, I was lucky enough to get a normal adult frog from these kinds of experiments. And I mention it because this frog, the first cloned frog, was entirely normal. People will tell you that cloned animals are sometimes not normal. That can be, but they certainly can be normal. And this particular frog lived for about 20 years and had about 5,000 offspring, all of which were normal. And soon after that, cloning, as we now think about it, took place. In this case, one takes an albino white-colored frog tadpole, separates the cells, like I've said, do the nuclear transfer to non-albino eggs, and you end up with an albino clone. And that's what this one looked like. So these are adult frogs. They're albino because that's where the nuclei came from, and they're small because they're males. They all have to be the same sex. And you can graft skin between them because they're genetically identical. It was not a result of any great scientific interest. It was obvious to those who knew what was happening, but of course it attracted a lot of media attention. People said, well, maybe you can clone Hitlers and that kind of thing. So I mentioned it in passing because it never really answered any kind of scientific question at all. So one now gets to the critical experiment, and that is, can you take cells of a specialized kind, like skin or anything else, and make these experiments work with those? So far, I've really only talked about experiments where you take cells or nuclei from an embryo. We really want to know whether you can take specialized cells and do the same experiments. And this was the first one where that was done. It was actually the intestinal tract of a feeding tadpole. The yellow color is actually the food inside the intestine. And so one was able to do this nuclear transfer or cloning from the cells of this intestine into eggs, like I've described. And when you do that, you often get entirely normal tadpoles. Here is one from a fertilized egg, and here is one from a transplanted intestine nucleus. They look almost exactly the same. If you look at the eye of one of these intestine-derived tadpoles, it's an entirely normal eye. All of these few thousand cells are parts of a normal eye, but remember that all of them started life as the nucleus of an intestine. So it makes the point that it really is possible to bring about major changes in the stability of cells by transplanting the nucleus from such a cell into an egg. Now, sometime after this, the success with mammalian nuclear transfer took place with Campbell and Wilmot. It's quite interesting that this remarkable result of the sheep cloning took 40 years to develop after the early work with the frogs. One wonders why it took so long. Actually, the methodology is the same. And the interesting thing is that the people who were working with mice and so on never copied exactly what was done with the frog. When they did, as Wilmot and Campbell did, then they got this result. And that is the cloned sheep and its offspring. People will tell you that Dolly the sheep didn't live a full normal life. Actually it did. And by the way, why did they give it a name? Because Dolly is a good name. Everyone knows of Dolly the sheep. They said, why didn't we give our frogs a name? The problem was there were too many of them. We couldn't think of enough names. So Dolly has gone into history as the famous Dolly the sheep. And Dolly lived for 6 years. And that was a good life for a sheep. Sometimes sheep can live longer. And the reason that Dolly didn't is that it was so precious. It was 1 out of 273 nuclear transfer experiments. And they were afraid that if they put it out in the field where they like to grow, Dolly would be stolen and put into a museum. So they had to keep Dolly indoors in a kind of appropriate barn to protect it. And as a result of that, it got a respiratory infection and then died at 6 years, as opposed to some slightly greater length of life that it could have had. So that's something about cloning. Teru Wakayama, he and his colleagues have done, I think, a remarkable experiment called serial cloning. It works like this. You take a mouse and you take the cumulus cell, that's the cell that surrounds the embryo, and transplant its nucleus into an egg, like we've described. You grow this embryo to another mouse and take its cumulus cell and transplant that nucleus into egg of another mouse. And so you go on. That's why it's called serial nuclear transfer. And he's done this amazingly for 25 generations. That's about 500 cell divisions from the start, and that's longer than any kind of cell will normally live. So it means that the nuclear transfer, in effect, gives you indefinite rejuvenation. The cells don't age and they don't die. And furthermore, there's no evidence of cancer. It illustrates the remarkable ability of an egg to essentially rejuvenate part of the nucleus or part of a specialized cell and keep it in an embryonic form. That's the key part of this nuclear transfer procedure. Now, I'll say a brief word about the mechanism. We all want to know, what is it that an egg has in it that can rejuvenate parts of a cell? It is necessary in these experiments to inject a nucleus of the cell, not the whole cell. That's why the egg substances can get into the nucleus and bring about the rejuvenation. We have some idea now of the molecular events that accompany this rejuvenation. And you'll notice that the times are short. These are hours after the cloning procedure, 3, 6, etc. This is the step I've just mentioned. A particular molecule goes into from the egg into the nucleus, and then there are various changes. This is replaced by another kind of histone, and this is replaced by modification of the surface of the histones. And then, interestingly, there's an actin polymerization. That means things can move. Possibly that's because that enables the nucleosomes to be rearranged. And then at 24 hours, this is not a lot of time, already there is now the reading of genes from the implanted nucleus, which has now been, to some extent, rejuvenated. That's something about the cloning. So I'll just give you the conclusions before moving on from this part of the work. These are the conclusions from single nuclear transfers to eggs. The first really important thing is that nearly all cells in the body have a complete genome. They have all the genes you need. And remember, that wasn't clear in the 1950s when these experiments were started. It's a fundamental point, and as I say, luckily it is true. Might not have been, but it means that every cell in the body, in principle, has the ability to form every other cell in the body. Second conclusion is that egg cytoplasm, which is the egg, has a remarkable ability to reprogram gene expression back to an embryonic state. That's to say, if you start with skin, the egg can turn the skin nucleus back into an embryo nucleus, make it start life all over again. Importantly, as cells differentiate, as they become things like skin, etc., the nuclei become increasingly resistant to reprogramming, so the success rate goes down, but nevertheless, it can happen. So now I'm going to talk about embryonic stem cells, and out of interest, how many of you have heard of embryonic stem cells? If you could put, well, almost everybody has. It's amazing, right? The question, of course, is, do you really know what they are? And perhaps you do. But in case you don't, I'd better tell you about them. First of all, what is a stem cell? We can illustrate this by skin. You have stem cells in your skin, and these stem cells will divide. When they divide, they give rise to one daughter cell, which is another stem cell, and the other daughter cell becomes mature skin. This is the cells which are falling off your skin all the time. And this cell never divides again. That's the end of its life. This one, however, can divide again, and when it does divide again, it makes another stem cell and another differentiated cell. And this just illustrates the skin stem cells. This is skin. Down the bottom is a cell which you can't recognize. That's a stem cell. It will sit there and go on producing mature skin. It happens in the blood as well. Your blood is always being replaced by the activity of stem cells. In fact, you may not know that at the end of this talk, you will have lost about 1 million blood cells. And it won't help if you run for the door, because you'll still lose them anyway. But this is the rate at which your cells are being reproduced, an astonishing rate, and that's due to the stem cells. But the really important key discovery was the embryonic stem cell. The real genius who discovered this was Martin Evans, who was working about a mile away in the genetics department in Cambridge, and he discovered these embryonic stem cells, and those are absolutely crucial. So here is an experiment where they took a monkey, skinned the monkey, and did the cloning that I've mentioned. The skin nucleus is put into the egg of another monkey, and this cloned nucleus and egg will make an embryo, like we've indicated. The embryo has cells in it which will form the animal, but if these cells are taken out and put into culture, they make what is called embryonic stem cells. And this was the key discovery of Martin Evans, made in about 1980, for which, very appropriately, he received a Nobel Prize. So we have monkey skin turning into beating heart cells, illustrating the fundamental principle that you can achieve this. Now, that's a large part, you might say, of the replacement problem addressed. Not quite all, because you have to find a way still of transplanting this beating heart cells from the laboratory into the patient. They don't like to do that very well. They get lost and don't beat regularly, but it's still a very impressive principle that you can, in this way, derive beating heart cells from skin. And these are now the embryonic stem cells, and they, importantly, remain embryonic forever. They don't make skin and so on unless you arrange it, but you can grow them indefinitely, and that's absolutely crucial. So this is a permanent reserve of stem cells which can form everything. And when you have these cells, you can add factors, molecules to them, and they can then be made to form brain and heart and so on. That's the essence of these embryonic stem cells, going back to the picture I showed at the very beginning of the talk, where we did cloning and took cells and made heart and brain. That has to be done making use of these embryonic stem cells. I'm now going to move to the next major phase in this whole work. This was the work of Yamanaka in Japan, and the problem with the work I've mentioned so far is that it's very difficult to do these cloning experiments with humans because you would need human eggs. And women, quite understandably, are not very happy about donating large numbers of eggs for experiments. And what Yamanaka and Takahashi discovered is that if you take ordinary cells, embryo cells, the kind of cells that Martin Evans made, and you add components to them, occasionally they will switch into embryonic stem cells. This is the kind of design that he used. So here is skin. You separate all these cells in a test tube, and then you treat these cells with particular genes. You can do that. You can grow genes, and added these to those cells. Occasionally, 1 in about 1,000 will suddenly flip and form an embryonic stem cell. Most will not, but here's one that has. You isolate that and grow these cells up, and so you get a whole collection of embryo stem cells without having to use eggs. And this procedure works in the mouse hair, works in humans. So here you can derive a nerve cell from the skin and can transplant it back to a host. So that's the principle of this amazing discovery of Yamanaka and Takahashi, which has really hit the whole field like a tidal wave. Thousands of labs are all trying to find out how to make this work. It works, but very badly, and it would be better if it worked better. But there is enormous progress, and in principle, you can now take skin or indeed any other part of the body and derive brain, heart, or anything from it using this very good, very splendid procedure. Now, just to conclude this part, these are conclusions from the so-called induced pluripotency stem cells, the ones I've just been talking about. These cells can create embryonic stem cells and hence all other kinds of cells from skin without the use of eggs. That's a key discovery. And as if that wasn't enough, these cells can be taken from diseased patients. And so you can grow cells from patients with, say, neurodegenerative diseases in the laboratory, and that means it's possible to test millions of compounds to see if you can find any chemical which could alleviate the diseased cells, Parkinson's, etc. You could never do that with humans because you can't test chemicals on living humans, but you can if you turn the diseased cells into laboratory cell cultures. So there's a great future for that. Finally, on this point, the most recent advance has been, as I call it, a look into the future. From cells to organs, a small piece of human skin can be used to make a continuously growing culture of stem cells, like I've mentioned. These cells are grown in dense cultures, and they eventually form three-dimensional clusters. And after several weeks in culture, some of these cells arrange themselves spontaneously into complex configurations which resemble those of adult organs such as brain and eye. This is still, of course, current work and has to be developed a lot further, but that is a possible future. And here is such a, they call it an organoid. These cells, these are thousands of different cells, but they're making patches. Cells of like kind stick to each other, and this bears a certain resemblance to part of a brain. So maybe that's the future, grow really bits of brain from tissues like skin. Last part of what I'm going to say has to do with whether this idea of cell replacement can actually be clinically useful. Nobody really wants to know how to clone a frog, but it would be wonderful if this instability of body cells can be used for cell replacement. And I'll illustrate this principle by referring to the disorder called age-related macular degeneration. Many people of mature years suffer from this, maybe 10% of those over 80 will have serious problems with this disorder, which eventually makes you blind. There is, in fact, a so-called dry form. The wet form is alleviatable, the dry form is not. This is the one which most people suffer from. So what can you do about the progressive blindness which will occur after the macular degeneration in humans? The eye has the lens, and behind it are all the relevant parts of the eye. Most importantly, there is the retinal layers with photoreceptors. This is the pigmented retinal epithelium. It's possible to implant these derived eye cells, even if you make them from skin, into the back of the eye. And this is surprisingly effective. The way it works is this. Here is a view of someone with the age-related macular degeneration. You see the central vision becomes completely obscure, so they can't see anything in the center. They have some vision around the outside, and eventually the outside also becomes non-functional, and patients like this become blind. That's how this disorder works, and it affects quite a significant number of people. Now, what can be done is to replace the relevant part of the eye. These are the so-called rods and cones of the photoreceptor cells, and they're in close contact with the retinal pigmented epithelium underneath. In the diseased form, the retinal pigmented epithelium cells die, and soon after that, the photoreceptor cells also die, and so you can't see. Now, what is done with this technique is to use the procedures I've mentioned to make layers of replacement retinal pigmented epithelium. Here are the replacement cells, and you can grow those in the laboratory in a little sheet of plastic and implant them into the back of the eye. So these replacement cells are now substituting for the diseased retinal pigmented epithelium cells, and this now rescues the further deterioration of the rods and cones of the photoreceptors. So you stop things getting worse, may even actually improve them. This is now the work of Peter Coffey from London, and there are other laboratories in the world doing this. Here is a sheet of these cells. These are the retinal pigmented epithelium cells as a monolayer. It's about 3 by 6 millimeters, and that can be surgically introduced to the back of the eye. Not a very complicated procedure, as a matter of fact. Here are the monolayer of cells, human ear cells to start off with, and turn them into the eye cells. And I should say this is working for clinical rescue of blindness with the eye. It does not yet work with the heart or the brain. And the reason is this: the human, like all of us, consists of 10 to the power 13 cells, huge number of cells. Human heart is 10 to the 11 cells or more. A bad heart failure will probably kill off about 10 to the 11 cells. Now, with the eye cells that we're talking about, you only need 50,000, a relatively small number. And this number of cells is sufficient when implanted into the back of the eye to rescue the rods and cones of the dying cells. So this procedure is fundamental because it shows you can actually do cell replacement in a clinically useful way using these cells for the eye. So let me now summarize my talk. I started by saying that all cells of the body have the same set of genes. If that had not been true, this replacement procedure could not have been used. Adult cells of one kind can be reprogrammed either through embryonic stem cells or even directly to cells of an unrelated kind, skin to brain, etc. And the replacement of some kinds of aged or non-functional cells in humans is a real prospect in the near future. That's the end of the talk. Thank you.
I think there's possibly just a little bit of time for some questions just at the front here.
I was wondering if you could give your thoughts on the possible limitations of the technology in traumatic brain injury or something, how much can you hope to regenerate in such a complex organ?
Well, it's a very good question. Well, what about the any repair of the brain or part of the nervous system? The cells that I've talked about do rather easily turn into brain cells. The problem is to turn those cells into something which can actually be inserted into the brain. But I think there's good prospects. I mean, it's rather remarkable that skin to brain cells works remarkably well, and they're physiologically normal brain cells. So there's a lot to be done to try to find out how to get those brain cells in the laboratory to actually integrate functionally into the brain.
I think there's possibly time for 2 more questions. So maybe a lady just here.
Because you mentioned that the cells become young again when you transfer the nucleus from a cell from the skin to the embryonic cell.
Yeah.
But those nucleus are already carrying mutations because
Oh yes,
they
I
are
see.
all
I'm sorry.
nucleus
Yes, no,
in
I've got the
some
yes.
way.
Yes, it's true that all cells of the body have some mutations in them, and they could be harmful. That's of course the mutations may be irrelevant to what you're trying to do. Supposing there's a mutation which affects, shall we say, the pancreas, it doesn't really matter if your eye cells have that mutation in them. Nevertheless, there is that possibility, and there's always the opportunity for testing the cells before they're implanted. The ability to determine the sequence of genes is getting increasingly effective. So it's perfectly possible, if there's doubt, before you implant cells, to test them to see there aren't any harmful mutations in them. That would be entirely possible. So I think it gets over that concern.
And one final question, maybe the lady in the stripy top.
You were mentioning the 25 generations of cell regeneration. Does that mean that this sort of thing could have applications in counteracting aging?
Yes.
And in the whole sort of industry of trying to counteract aging?
Yes. Well, it's a very good question. The 25 generations, which I mentioned, I mean, point is, could that counteract aging? I think we will really have to know what it is the eggs have in them that do give this kind of indefinite life. We don't know that yet. That's really what I think is going to be needed for that. Current work is, of course, aiming in that direction. Otherwise, I don't think it has a likelihood of, it's not going to make any of us immortal.
John has mentioned many of the other people who have contributed to this area of work, but hasn't mentioned himself. And of course, he's been very involved in all of this work for a number of years. So I'd like to thank him for his time today and all of his amazing work that's going to contribute to health in the next 25 years, I'm sure. Thanks very much.
Yeah.